Transcription is a process that creates mRNA, and it starts in the nucleus of the cell. It all starts with a strand of DNA, which holds the information that will ultimately be used to create proteins that are vital for survival. In the DNA, we see that there is something called a TATA box, or a TATA box. This box lets the RNA polymerase II know when and where to start reading the code in the DNA. There are 3 different types of RNA polymerase. There is RNA polymerase 1, 2, and 3. I'll tell you about what they do as we come across them in the protein synthesis process. From the code in the DNA, the polymerase II makes a strand of nucleotides called mRNA, or messenger RNA. This strand may look familiar, as all the nucleotides match up to the same nucleotides that they did in DNA, except for adenine, which pairs with uracil instead of thiamine, like in DNA. RNA polymerase stops creating the mRNA once it comes across a stopping codon, which is a set of 3 nucleotides that lets the RNA polymerase know when to stop. In this example, the stopping codon is UAA. Once the RNA polymerase II is finished creating the mRNA strand, the mRNA then ventures outside of the nucleus to either the endoplasmic reticulum or to the free ribosome, which is made of ribosomal RNA and protein. The RNA is created by polymerase I in the nucleus by using rDNA genes. The free ribosome uses the information from the mRNA to build proteins in the next step called translation. Translation happens in either the endoplasmic reticulum or in the free ribosome. In this case, it happens in the free ribosome. Once the mRNA enters the free ribosome, tRNA, which is made by RNA polymerase III, connects to the codons, which are sets of 3 nucleotides, on the mRNA strand. Every tRNA has a different amino acid. Once the tRNAs find and connect to their respective codons, then the amino acids at the top are in the right order to bond and form a polypeptide chain. The chain is then broken off by a stop codon, which in this case is UAA. These polypeptide chains are what proteins are made out of.